TV screen, so the images are being created from the data that's being generated inside the microscope. So there's no eyepieces to look at anything. You're not looking at things. So here are your images being projected. We are looking at two different modes, because we can have multiple detectors going on, because we can have our secondary electrons being captured and creating an image, and then we can have a different detector at the same time elsewhere in the microscope collecting the X-rays, giving us an X-ray image, and then we can have a third detector collecting something else, and we can simply, all these events are taking place at the same time. So if you have a detector, you can capture as many of them as you want. So let's look at this image here. Should look familiar for you.
This stuff is going to be in the exams for next week too? The demonstration stuff?
Just in a general way. So this looks like a typical transmitted image. Your bars are blocking the beam, so they're dark. Your plastic here doesn't have any sample in it, so it appears light. And these are these individual cells I was describing to you that look like swimming artichokes. By comparison, if we look at this image, this one is showing us the grid bars, and you're getting this three-dimensional information coming off the grid bars. And then in the background, you can see this is your slice. You can see that it's on the other side of the grid relative to the beam coming down the column, because you don't see any evidence of it here on the grid bars.
Is this the smooth side or rough side of the grid bar? Of the grid?
I don't remember.
Okay.
I should have some of the students in the class look at it next semester. But this one, it looks more like the rough side.
Okay.
And zooming in, as I say, you can see. Let's see, we need to go to a faster scan by having a smaller area. And so, what is this image telling you as I go through focus here? Stigmatism, because you're getting this bad distortion. So this picture right there is your best focus, but we can try to improve that by fixing the astigmatism. And now as we go through focus, it moves, but it's not distorting as much. So I've managed to correct the astigmatism to some extent, and I could spend more time with it, but that's not our purpose for being here. So we're more interested in the cells. Yes, am I missing something? So these are the cells. So here's where I just popped a hole in this grid.
Is that due to the bending?
No, this is our accelerating voltage, so it's too much energy. And I think I'm too close to the grid, so I think there's some heating going on. So here you can see this is about the best image I can get for this particular set of settings. If I reduce the size of my spot, we should get a better resolution. Making that change just really messes up our stimulators. So this here is the edge of the Golgi apparatus. Here you can see this is cytoplasm out here. This is one of the chloroplasts. And here you have these vesicles inside the cell. So this is down near the side of the cell away from the flagella. And so these are now ready to be secreted because these look like fully formed scales inside here. So here you see the scales. They look like sombreros out on the outside of the cell body.
This is scale. You keep saying, but scale to me is the thing that you go and weigh yourself. It's not that.
Like a fish scale?
Right. And you have fish scales.
Like at the top of the body.
Yep.
Oh, okay. Got it.
And they can have dramatically different shapes. Some of these cells can have multiple layers, and each of the different layers can have a different morphology. One layer can look like a spider web and very flat and spider web, and another layer next to it can look like literally like a crown that you would have from a queen or king. Big tall thing with spikes coming up out of it and everything. All of them are produced in these vesicles inside and secreted. And different groups of these single cell plants have different compositions in their scales. So these scales are silica. In another group called coccolithophorids, those scale-like things are called coccoliths, and they're the ones that make up a large component. Some of them make up a component of things like the White Cliffs of Dover, are the residuals of plant cells. And those can look also like very flat cells. I don't know if I've got any pictures of those. Let's see, what are some different pictures I might have available? So these are probably coccolithophorid scales. And these were ones created inside. This is that poor cell. So these were secreted to the outside. These are made of calcium carbonate. And the ones that we're looking at in this other organism are silica. In the green cell, in the green plants, in the cells that are green, their scales are organic. But they're all, again, being produced inside these vesicles and secreted. And again, nobody knows how it controls accumulating and then depositing these materials, how the orientation of how those microstructures and everything are determined. And that's, as I say, a great mystery involving microtubules, microfilaments, and who knows what else. And it's genetic because it's different organisms produce different shapes and they're constant. So when you see that shape, you can say, yes, that's in this group, and identify it down to maybe a genus or a couple of different genera. So it's, as I say, something that's got to be reproducible. let's see. If we look at something like so this is trachea. This is trachea from a mouse. And so these are the cilia in your ciliated epithelium here in the trachea that move things out. Rumor has it that when you're a smoker, these hairs are burned off or damaged so that they're no longer functional. And that's why smokers have a harder time clearing their lungs, and supposedly that's why you have more junk inside of smokers' lungs than non-smokers' lungs. Some physiologist came through when I showed this slide, and she said that supposedly when you're a pot smoker, the hairs don't grow back if you stop smoking the way they do with tobacco. So if that's true, then when Clinton dies and they look at his trachea, we may be able to find out if he actually inhaled or not.
President Clinton, he was a few years back, several years back, and
You see, he's addicted to the drug or something? I don't get the joke.
Clinton was accused of smoking pot, and he said he never inhaled pot. But that was like some of the other things he said he didn't do.
We won't go into detail about those.
We won't know. So if it's true that if you are inhaling pot and it burns off the hairs, they don't grow back, then if they do an autopsy, they shouldn't find cilia in his trachea.
I got it.
This is diatom, so you can see how different this looks from that one that's out on the wall. And again, this is highly reproducible. So as I say, whoever can figure out how they do it in the backyard in the bucket is going to be sitting pretty. Let's see.
did you find any of those in the lab yesterday, Danny?
Yeah,
Yeah, me too.
centric and a pinnate.
These are, so this is one of the modal cells I'm most interested in right now. This is naked, it doesn't have any scales on the surface of it. It's got two flagella, and then it's got this third thing that looks like a flagellum, but it isn't. And it's thought to function in capturing things like bacteria that it then brings to it, and once it gets close to the cell body, it sort of touches the cell body, and the cell body sucks it in. And they're not quite sure how it does it, but there is some evidence of seeing bacteria inside of some of these cells, which suggests it's actively taking it up and breaking it down to survive. So even though they're photosynthetic organisms, they also have this ability. They're also phagotrophic. They can eat things and survive on that. So this would be its form when it's all coiled up, and when it's extended, it can be 3 times the length of these flagella, so it can be quite long. And it swims. One form of swimming is to have this thing extended either in front of it or behind it, with the 2 flagella off to the side, so it kind of floats with this thing sticking out. See all these things you won't learn anywhere except talking to an algae person like me. All right, so here we are hoping to identify what these scales are made of. So we can go here and take a picture. All right, so let's create a new picture. It's as slow as molasses. All right, so we we can do a little post-processing here. See what a difference it can make. All right, so now we can go in and we can either map this, or we can simply say, I want to take a point here. So let's find someplace that's chock full of silica, and it's not going to collect X-rays from that single point right there. And so here you can see we're collecting silica, and we can spread this curve out. And we talked about how images can be very noisy. Like when you are looking on the TEM camera and it's moving around, the image isn't really crisp like it is when you take the picture. So when you're taking your picture, you're taking a multiple scan, averaging them all together kind of a thing to get rid of the snap, crackle, pop. In this case, our snap, crackle, pop in the electronics shows up as this giant peak here. So if we can tell it to remove that, you can see that it's now reduced and the scale here went down. And so we got oxygen, aluminum, and silica. I don't know where the oxygen is, the aluminum is coming from, but the holder has aluminum in it, so it's possible that it's coming from that. But the holder, here's the holder here. So this is the holder. So you can see it can take 5 or 6 grids, and they all have little flaps that are going to hold it by the skin of its teeth, so to speak. So it's just going to hang out there.
Each of these ones?
Yeah,
Yeah,
so
each
it's
one
like
of
a
those.
clip.
It's-
So it like clips the edge of it.